There's an old joke that floats around the internet, and it goes like this. Biology is the only science where multiplication and division mean the same thing. Ha! Okay, you may be wondering, why is that funny? And the short answer is, it isn't funny, it's educational. Ugh, shudder. But wait for a second. What actually is cell division? I want to take a moment today and talk to you about mitosis, because it's actually very interesting. So for starters, we should understand that cells need to replicate. You can thank cellular replication for the time you grew 4 inches overnight in middle school, or whenever your fingernails get super long and you put off clipping them for probably a little too long. The reason your body grows the way it does is thanks to cells. If you think about it, it's obvious. Of course, the amount of cells in your body needs to increase as you get bigger. What's not so obvious is the cells also replicate to replace each other. If you've ever experienced a particularly nasty sunburn, you've experienced this firsthand. Have you ever wondered what a sunburn actually is? I'll tell you right now, it's not your skin cooking like meat in a hot oven. It's actually the cells in your skin performing a special routine known as apoptosis. Apoptosis is the fancy science jargon for programmed cell death. So in the case of a sunburn, your skin is exposed to the harmful rays from the sun, which are known to damage and sometimes mutate DNA in your cells. So to prevent your skin from becoming cancerous, your cells decide it's best to kill off a layer of skin cells and replace them with newly replicated ones, which are less likely to have damaged DNA. It's kind of wild to think about. Some cells replicate more than others. For example, neurons, which are computational cells common in the brain, and the cells in your heart replicate very, very little. In your heart, cells are said to only replicate during growth phases, such as when you're a developing fetus in the womb. They don't replicate much else than that, though. In comparison, some cells replicate quite a lot. For example, the cells responsible for your hair replicate every 1 to 3 days. When we talk about the replicating cells, we usually say the cells divide. For example, this cell here is about to divide. The one cell will become 2 fully operational daughter cells. Isn't that beautiful? The question you need to ask now is, how does this happen? Enter our big word of the day, mitosis. The technical definition I like to use for mitosis is this: the process in which a cell splits its nucleus and distributes a copy of DNA to both nuclei. Before we get into all the nitty-gritty of what that means, let's talk about mitosis and what it is and isn't. Contrary to what you might be told, mitosis is not a set of clearly defined stages, so much as it is a series of changes that we arbitrarily differentiate from one another. I'll try to include a link in the description to a video that shows the mitosis process in action. If you watch it, you'll notice it's a very fluid process. There's no resting point. I'm going to point out all the little details of mitosis, but as I do, keep in mind that all of these phases of mitosis actually happen very, very quickly in conjunction with one another. When a cell divides, it starts with 1 nucleus and ends with 2 identical nuclei. This is important to remember because the DNA is held in the nucleus. It's important that the DNA of the mother cell is identical to the daughter cell so that the daughter cell may perform the same role as the mother cell. We call this outcome cloning, and if you ever hear someone refer to cells being clones of one another, this is what they're referring to. There's one more thing I want to make very clear before we dive deeper into the actual stages of mitosis. Although all cells divide, not all cells divide using mitosis. Some very important cells use meiosis instead. We'll talk about meiosis perhaps another time, but for now, you should just be able to recall that sex cells, those that are our sperm and our egg cells, they do not use mitosis. Instead, they use meiosis. It's a bit different, it's a different process, so keep that in mind. So let's jump right into the steps of mitosis. We'll start with the first phase, prophase. The pro in prophase means before, just like in the word proactive. In this phase of mitosis, notice the nucleus starts out just chilling. It still exists. Towards the end of prophase, the nuclear envelope that basically separates the nucleus from the cytoplasm altogether fragments. This is important because the cell wants a way to sort out all of those chromosomes that are stuck inside the nucleus and let them loose so that they can be mingled with by the rest of the cell. Okay, the other thing to notice is the chromosomes in the nucleus. Now is a good time to talk about what happens and what will happen to these chromosomes. So before mitosis even starts, the DNA in every single chromosome is copied. What's cool is the amount of chromosomes in every cell, 46, remains the same before and after DNA is copied. So what happens? Well, those chromosomes just double in size. We say that this half a chromosome is actually a chromatid. So here's a set of chromosomes, all of which consist of one chromatid each. Then a DNA is copied, and presto, the chromosome count stays the same, but the chromatid count doubles. Keep in mind that the chromatid count actually doubles before mitosis even starts. And before we start prophase, the chromosomes don't look like this. They don't look nice and ordered. The DNA that makes up each chromosome is actually more like spaghetti than it is tight-laced. But once we hop into prophase, those chromosomes are going to tie their bootstraps, and bam, the chromosomes condense to form less spaghetti-looking mess and a more rejected Lucky Charm shape that we are all familiar with as the classic chromosome shape. We call this process condensing. The last two important things to mention in prophase have to do with those centrosomes. Centrosomes are a crucially important body in the cellular division because, as you see in prophase, proteins known as microtubules attach to these centrosomes, and then each of the two centrosomes in prophase begin a long and arduous migration towards opposite sides of each other. Sometimes you hear their centrosomes called spindle poles. Just like the Earth, which has two poles on opposite sides of each other, a cell in prophase has two poles marked by those centrosomes. Now, the next phase of mitosis is sometimes omitted from the classroom, but we're going to talk about it nonetheless. It's called prometaphase. We can easily remember the term prometaphase as this phase is sandwiched between metaphase and prophase. Meta means middle, and again pro means before, so this phase is literally called before the middle phase. Cool, right? So what happens in this phase? Well, we'll see two main events. For starters, we'll see some of what we saw in prophase carry over, like the chromosomes condensing. We'll also see the microtubules attached to the chromosomes, effectively tethering each chromosome to both centrosomes. But wait a second, how do the microtubules just attach to these chromosomes? That's kind of insane if you think about it, how they just do that. Well, the cool thing is that microtubules attach to a special protein located at the center of the chromosome, called the centromere. They latch onto this centrosomal region by connecting to a special set of proteins called a kinetochore. These proteins are cool because they function a bit like a loading dock, for which the microtubules can bind to. The next phase of mitosis is metaphase. In metaphase, a few key things happen. Metaphase is going to pick up right where prometaphase left off. Now that the microtubules of each centrosome latch onto each of the chromosomes in prometaphase, metaphase can move around the chromosomes freely. Now the hallmark of metaphase is that these microtubule spindles begin to arrange the chromosome pairs towards the middle of the cell. If you were ever asked what one of the straightest lines in nature is, although contestable, a good answer might be the metaphase plate. The metaphase plate is the straight line here, made up of specially positioned chromosomes. We'll see why the cells arrange their chromosomes like this in just one moment when we move on to the next phase. The next phase, of course, is anaphase. In anaphase, this metaphase plate we just saw our cells work very hard to create is going to get torn into two halves. We can easily remember this happens in anaphase by remembering the prefix ana means to make things part. Think of the word analogy, where two things are similar but still apart. Well, here in anaphase, the metaphase plate parts into two identical halves. So as this metaphase plate is being split in two, the spindle poles, remember, are centrosomes, they're going to migrate to polar ends of the cell. And you might be thinking, wait a second, didn't the spindle poles already do that, like way back in prophase? And if you're thinking that, you'd be very right. In anaphase, this movement of spindle poles actually corresponds to the elongation of the cell as a whole. Imagine two positively charged magnets repelling each other in a balloon. That's kind of like what's happening in our spindle poles and the cell body as a whole. They're behaving as though they want to get as far away from each other as possible, and in doing so, the whole cell is stretching. And as the centrosomes migrate away from one another, those microtubules, those spindles that attach the centrosomes to the chromosomes in the metaphase plate, they get yanked. And just like a wishbone getting snapped in two, each chromosome promptly breaks into two equal halves, with the breaking point being located at the centromere. And then the next phase of mitosis is telophase. Now, the prefix telo means something like end, and that's kind of what we see happen in telophase. Technically, telophase is the last stage of mitosis. Although we certainly have more to learn, telophase is where the term mitosis meets its telo, its end. So what's happening in telophase to accomplish the end of mitosis? Well, a few things. Number 1, let's remember where we left off in anaphase. Our centrosomes were migrating away from one another, while in telophase we have the chromosomes themselves on opposite poles. Remember, the microtubules get yanked, and so did the chromosomes with them. In sort of a grand action, the fragments in nuclear envelope we talked about way back in prophase recongregate to form and envelop the chromosome groups, so we get 2 new nuclei. Remember that the term mitosis technically means the division of nuclei, and those phases we just walked through, they all culminate in this, the penultimate phase, telophase. Now, before we leave telophase, it's worth mentioning that the spindle fibers, which accomplish so much in mitosis, in this phase, they break down. No longer are they very active in this process. So we've talked about the 5 main phases of mitosis. The next phase we're going to talk about isn't so much a phase of mitosis as it is an event of the cell cycle onto itself. It's called cytokinesis, and it just means the division of the cytoplasm. This happens in close conjunction with telophase. A cleavage furrow will form towards the center of the cell body while centrosomes are still pulling away from each other in telophase, and soon enough this furrow will grow to completely split the cell into 2. This is really the division that we talked about at the start of the video. Pretty cool, right? I think so. So there we go, the end of mitosis and cytokinesis, as 2 daughter cells identical to their mother cell. You'll remember that at the start of prophase, there is twice as much DNA than is in each cell now, because the DNA was divided between them, of course. Later in the cell cycle, should these cells decide to divide again, they will both double their DNA just like their mother cell did. It's the circle of life, or the cell cycle, I suppose. So cool. I hope you've learned a little bit more about mitosis and cell division than you previously knew. Happy studying.